Hi Gaurav, thank you for joining this webinar and contributing to this initiative. My name is Naveen Kumar and I am European Project Manager for Irish Bioeconomy Foundation. We are based in Tipperary, Ireland in National Bioeconomy Campus. This webinar is about our project. We are working on Rural Bio European funded project, which is focusing on technologies and business models in Europe on bioeconomy. And we want to convey those technologies and business models to the normal audience community and get this knowledge out. So I would like you to speak on something about like you are working on the different type of technologies you are working on, and I will let you speak first with your intro, and then after you can speak on your slides. I will turn off my camera now and we'll get back after. Yeah. Thank you. Please go up, continue. Thank you.
Thanks, Naveen. And also, Dr. Gaurav Rajauria. I'm a senior lecturer here in School of Microbiology, Food and Nutritional Sciences, and a PI at SUSPAM Center for Fermentation and Bioeconomy at University College Cork in Ireland. And I'm just practicing and researching in the area of biorefinery and biomass valorization from the last 15 years. So I have worked on a number of research projects from national funding agencies and European funding agencies. And my research group, which is Biorefine, which is actively working in the area of biorefinery development for feed, food, and platform chemicals and other ingredients production, utilizing different pre-treatments and fermentation technologies. That's what we do. And I am going to basically share some of the biorefinery development processes that we designed over the last, let's say, 4 or 5 years, and how we valorize different biomasses, how we produce a number of bio-based products that for food and feed sectors. So I'm going to share my screen and so I guess, can you please confirm that you can see the,
yes, well, I can
screen?
confirm. I can see your screen. Thank you.
Perfect. So as I said, so I just start with the basic idea of, I'm sure this slide is not moving. Yeah, it's moving now. So I always start with a very simple and the idea of biorefinery and why we need that, and what is the biorefinery concept. So as I said, like because it's the biorefinery is kind of facility that integrates, let's say, biomass conversion processes, different equipment, pre-treatment technologies, and we utilize different conversion processes, and we produce bio-based energies, food, and platform chemicals from these agri biomasses, or not only like any kind of biomass that is available on our planet. So for general people, like, what is biorefinery? So because it's the concept is exactly analogous to petroleum refineries, because in petroleum refineries you use crude oils and fossil-based resources, and you perform different kind of processes, and you produce fuels, chemical products, and materials, and then you of course release a lot of carbon dioxide. But as against the oil refineries, in case of biorefineries, of course, you do the exactly same process, but the processes, I mean, here, instead of using fossil-based biomasses, you use fossil-based, resources. Here you use natural resources, I mean, the biomass that is coming from agri-food and different sectors. And of course, the processes are mainly green processes that we use in biorefineries, and then we can make exactly the same product that you make in oil refineries, and then you certainly can replace the exactly same product through biorefineries. And the important part here is the CO2 that we generate. We also have a capacity to capture that CO2, and we can also feed that CO2, and we can further produce a different food and feed ingredients. So it advocates the circularity in the system. So in the end, we do not leave any kind of waste behind if we basically work on the zero-waste biorefinery. So the idea of the biorefinery is we have to utilize everything that is available in the biomass, and we leave no waste behind. And the important basically the advantage of utilizing biorefineries, because in the case of oil refineries, what you do, you make a certain kind of product, but in case of biorefinery it is very, very different, because in biorefinery, if you take the same feedstock, let's say if you take any kind of feedstock, let's say wheat straw, which is a one biomass, and if you modify different processes, you can make n number of products. You can make energy from the same feedstock, you can make platform chemicals, you can make feed, you can make proteins, you can make materials out of that. It's just you can tweak the whole process and you can make a variety of product, which means these biorefineries can be tailored. So that is the beauty of these biorefineries as against the oil refineries. Now talking about the process, so if we look at it, so there are some fundamental unit operations in any kind of biorefinery development, and there are some fixed requirement as well. So biomass, or I can say feedstock, biomass, there are different terminologies that people use, but biomass, feedstock, if we say, so any kind of let's say organic material that is available on planet that is coming from agri-food sector or different sectors, that can be used as a biomass. And of course, the primary unit operation of a biorefinery development is pretreatment, because all these biomasses are really, really hard, and in order to utilize them in to produce different bio-based product, you have to do some sort of pre-processing or processing. So pretreatment is basically how we can hydrolyze, how we can break down that hard matrix of biomass and convert into a hydrolyzate. So there are a number of, let's say, techniques that we use. There could be chemical techniques, there could be physical techniques, and a combination of both, and some biological techniques. And after that, once you hydrolyze that biomass, you then have to convert that hydrolyzate, if I can say that, which is, let's say, a degraded material, and you basically convert into a new product, or you can use that hydrolyzate to recover the product that are already present in the biomass. For instance, a biomass might contain proteins, polysaccharides, polyphenols, and other kind of active components. You can recover them, or you can use them as an intermediate substrate, and you can make a completely new product by using fermentation or precision fermentation approach. And in some cases, you can also use digestion approaches, where you can utilize the biomass and convert it into energy and biofertilizers. So in this second unit operation, you generally develop the intermediate product into a final product. And then, of course, once you make the product, it is not necessary that your product is in a pure form. It might contain a couple of impurities or residues from original biomass. So in this stage, you basically purify your product. You might need to concentrate if your product is in a liquid state or in a diluted form, or you have to perform different purification approaches and recovery steps in order to get your final product, and then you get your bio-based products. So that is basically a general approach of a biorefinery process, and these are, let's say, I would say, a major step that are that are involved in every kind of biorefinery development. There's one important thing here: in some biorefinery processes, pre-treatment and the conversion, sometimes they are a single process. Like, it is not necessary that you have to go with pre-treatment and then conversion. For instance, in case of extraction-based biorefineries, generally these two processes are combined, where you can use one of that process and then you can produce the product, and then you can do the downstream processing. So in some cases, pre-treatment and conversion, both steps are one step, while in other biorefinery process, they are two separate steps. That depends upon what kind of biorefinery you are developing. Now, as I said, the types of biorefineries. So there's I would say, a classification. So this image is not my image; it's adapted from another paper. So so we can classify biorefineries based upon the kind of biomass that are available around us. So let's say yellow biorefinery that is focusing on the side streams of biomass that is coming from our agriculture crops, or let's say wood chips, or wheat straw, barley straw. And if we are using any biomass coming from meat processing industry, we generally categorize it into a red biorefinery. And any biomass coming from marine sources is basically categorized into blue biorefineries, or any biomass that is coming from industry side streams. Let's say if any biomass coming from any manufacturing or processing unit, that goes into a gray biorefineries. And brown biorefinery is basically categorized any kind of effluent that is coming from, let's say, dairy processing plant or meat processing plant or any other kind of processing plant. The effluent that comes after making the product, and that effluent contains a lot of nutrients that if you are working on that kind of nutrients, it goes into brown biorefineries. And finally, the green biorefinery, so a lot of forest tillage, leaves and needles and different kind of let's say leaves and fallen leaves and let's say crop stock that is not being used, that if you are working on that, that is categorized into a green biorefinery concept. So based upon biomass, we can categorize into 6 different kind of biorefinery processes. And so all of that, because they all contain organic matter, so if we are using all of that matter and in different biorefinery process, that is basically a biomass sources. And of course, we have in Ireland, we have a plenty of that biomass, and this is very, very important if you are working on a biorefinery process, because if you are focusing on a large scale operation, so the first question that comes in your mind: do you have enough biomass that can be processed through a biorefinery? So you don't want to work on a biomass that has a seasonality. I mean, if you produce only once or twice, like in a particular season in a year, or if you are have a limitation of that biomass availability. So you have to basically work on that first if you are working on a biorefinery principle. And the best idea is if you are working on a biorefinery, you should have some sort of mixed biomass capacity in your biorefinery. So that will basically keep you going throughout the year if you are working on mixed biomass in your biorefinery development. Now, talking about the availability of biomass in Ireland, so this is the information that there was a mapping done by another research group here in Ireland. So they have identified 5 different, let's say, thematic areas or streams where number of different sources are available for primary biomass and secondary biomass, so the crop, forestry, dairy and meat industry, brewing, distillation, and cider making industry, and marine sector. So there are 5 different sectors that have huge amount of biomass availability in Ireland. So if you can give a little stats here. So roughly agriculture sector produce around 3 million tons of biomass that can be used for biorefinery development. Similarly, forestry sector has a capacity of close to 1.4 million tons of biomass that can be valorized into value-added product. And in case of marine, our industry and urban waste, we have capacity of 1.8 million tons. So all of that agriculture, forestry, and industry and urban waste that falls into a lignocellulosic category. But there's another category which is non-lignocellulosic, which is a little bit of a scientific term, but because of the presence of a specific compound lignin in the biomass. So we also have a capacity of 60,000 tons of biomass, which is non-lignin biomass that comes from marine sources. And I'm not including the grass biomass that is available in Ireland. So roughly we produce 30 million tons of grass biomass, and if we still have around 20% surplus, 20-25% surplus grass that can be used for developing different kind of biorefineries. So this is a landscape of availability of different biomass in Ireland, and of course, we have a huge, I would say, the capacity that in order to valorize these kind of biomass into value-added products. Now, once you have biomass, I will briefly talk about how we are going to hydrolyze it. So there are a number of technologies that you use, but the primary thing is why we need pretreatment, because this is, let's say, I would say, one of the costliest portion of a biorefinery development, and it costs around close to 20% of total capital investment if you are developing a biorefineries, because it's like different kind of biomass has different structures, and you need very, very different kind of chemical, physical, or biological forces to hydrolyze that biomass and produce intermediate hydrolyze it. So as I said, you need the idea of developing or applying pre-treatment steps or technology is basically you can destroy the matrix structure of feedstock, which means you can increase the surface area of particles, and you can also remove the cross-linked matrix that are holding different polymers and molecules, biomolecules inside the cell matrix. And you can also disrupt the crystalline structure of compound, which is, I mean, you can break down the polymeric structures and you can break down the crystalline structures. So all these polymers will be biologically available in the solvent that you are using during the pre-treatment. And also pre-treatment increases the mass transfer when you use different solvents in order to release those compound in the liquid matrix. And overall, it increases the rate of hydrolysis, so you can get a better hydrolyzate that can be processed further for conversion steps. So this is, as I said, this is a really important step in the area of biorefinery development. Now, talking about different technologies, as I mentioned earlier, so there are biological methods where you use different kind of enzymes and microorganisms that breaks down the biomass, and you can use different thermochemicals. So there are 4 methods: biological, thermochemical, physical, and chemical processes. And every process has number of methods and technologies that you can use. And based on these all, the selection of these approaches or technologies is basically based upon what kind of product you are going to make, whether your product is heat sensitive, light sensitive, or it is a heat stable product, whether it is with a harsh chemical is needed or a mild chemical is needed. So based upon number of parameters, you use different kind of extraction or pre-treatment technologies, and then you basically hydrolyze the biomass and you get the hydrolyzate. So this is an intermediate, I would say, a product that is produced, and then from there you can go whether you want to recover the existing compound that is available in the biomass, or you want to make a completely new compound through a fermentation process. So then you decide after this step. Now, moving forward, once you, let's say, get the hydrolyzate, and let's say if you are going for a fermentation approaches or if you are going for extraction approaches, you can produce your product, but that product might be, let's say, concoction of different things together with your compound of interest. So you have to recover those compound. Basically, you have to purify that. So there are a number of technologies that you use, and again, that is based upon the product that you are working on. Whether if you are using a fermentation approach, your product might be in the broth, which is extracellular product, or if a product is within the cell wall of microorganism, that is intracellular product, or microorganism are itself as a product. So based upon the location and of the product, as well as different impurities that are present in the along with the product. So you basically pick your downstream processing approach, whether you need cell separation or cell disruption approach, or you need filtration, what kind of filtration you need, and centrifugation, concentration. And once you get that, then you have to check the purity and purity and concentration of that product that you are recovering. And once you get that, then you have to finalize whether you have to dry the product, or you have to lyophilize it, or you have to use a spray drying, or you have to use let's say a hot air drying. So based upon your end product requirement, you pick different steps or different technologies. Again, it is not like necessary that you have to use all of them, but sometimes you need more than one approach in order to get your final product. So in this step, you get a pure and soluble and a final product and for ready for market. So that is what is basically done in the downstream processing step. So these are the major steps that are included in the biorefinery development. So probably my focus today is, I mean, not only the fermentation, because I am working on extraction based biorefineries and fermentation based biorefineries. So I give you two examples. But before that, I just want to give a little bit of idea of fermentation system, because a lot of groups are working in the area of precision fermentation, and how precision fermentation is helping to valorize these biomass and developing new product. So like there's a biomass fermentation, there's a precision fermentation. So there's a little difference here. In case of biomass fermentation, we just take a microorganism. We do not modify that microorganism. We do not genetically modify it. We just take the biomass. We take the food grade or grass status microorganism. We perform fermentation, and we produce product. While in the case of precision fermentation, where comes the synthetic biology and strain engineering. So in order to, let's say, make a product that in a larger quantity and a very specific product, you tweak the genetic profile of microorganism, and then you instruct the microorganism to produce a specific compound that you want to make. So based upon that end product, you tweak the genetic profile of microorganism. And then, because there are huge number of, we have a huge microbial biodiversity, so you pick the right microorganism. That could be yeast, bacteria, or fungi. And of course, there should be a grass. They should have a grass status. And then you perform synthetic biology and strain designing, and then you take the different biomass as a carbon source, and together you put all these things, both, I mean, the genetically modified. I generally don't say genetically modified, but I say engineered strain of microorganism, and then you use feedstock. Basically, you take sugars out of these feedstock of fermentable sugars. Together, you put both things together into a fermentation-based biorefineries, which is a fermenter or bioreactor, and then you optimize certain parameters, and then you can produce various product that could be green energy in the form of bioethanol, biodiesel, and bio-based chemicals. They could be any kind of platform chemicals, lactic acid, polylactic acid, that can be used for polymer development, bio-based materials that you can make, and composite design. You can do it by producing product here. And you can also make different food and ingredient, like you can make single cell proteins, microproteins, bio oils, and that can be used. And you can also recover the pigments, and you can also produce pigments that can be a colorant that can be used in food and textile industries and other sectors. If you look at it, the contribution of fermentation of biorefineries in different United Nations sustainability goals. So you can see simply, if you are focusing on green energy, utilizing bio-based refinery, you are targeting SDGs 7 and 13, where you are reducing the greenhouse gases by bringing green energy. And if you are focusing on bio-based chemicals and bio-based materials, you are reducing the use of fossil resources. And if you are working on food and ingredients from these biomasses and biological resources, you are basically focusing on UN SDG 2, where you are feeding the population. And by utilizing different different feedstock, you are also focusing on to reduce pollution in the area of SDG 14 and 15. So that's how you are. If you are working on a biorefinery, you are basically you are working towards at least 4 or 5 different UN SDGs. Basically, you are working towards reducing the impact of our carbon footprints through these systems. So that has a huge positive values towards the climate sustainability. So I'll take you I'll basically give you one example of I mean, my first case study is on how to develop a biomass to protein biorefinery. So this is the project that we are working on, funded by Research Ireland. So in this project, we are developing a microbial protein or single cell protein, utilizing different kind of biomasses that are available in Ireland. So this is, let's say, the idea that we proposed was, so we were focusing on different food or let's say agri-food side streams. We were focusing on food processing side streams, and side stream from the brewery industry, which is brewer spent grain. We are focusing on spent mushroom and vapor meat that is coming from dairy industry. So we picked the biomass that are abundantly available in Ireland. Then we are working on, and we developed a process that can this is a pre-treatment process where we are utilizing like ultrasonication, we are utilizing microwaving, we are utilizing hydrodynamic cavitation in order to break down these matrix and produce a fermentable sugars out of these biomasses, because all of these biomass contain huge amount of different sugars and the aim of that pre-treatment was to recover those sugars that can be used as a carbon source for growing our microorganisms. So we have selected different microorganisms from yeast as well as from fungi category. They are food grade microorganisms. And then we grew them into ferment. So we optimized different fermentation parameters and we brought using microbial fermentation. And then we produced our protein, and then we performed different filtration approaches. And then the protein was dried using spray drying and freeze drying. So this is the let's say schematic of our biorefinery that we are working on. And if you look at a little bit deep how we did it, so as I said, the primary step is you have to select the microorganism, which could be, let's say, there's certain criteria that you have to use when you are selecting the microorganism. First, it should be a food grade. So it should have a grass status, and it should basically it can grow on a large number of biomass because, as I said earlier, in order to make your biorefinery process more sustainable, they should grow on a number of biomasses. So you will not have an issue of biomass scarcity in your process. So in our process, we picked two different fungi, Aspergillus oryzae, which has been used widely for making rice koji in Japan. So it is being used for food fermentation in Japan, which is a food grade microorganism. We also use Pleurotus austriatus, which is a mycelial fungus, and again, it's safe to use, and it has extra added advantage because it contains a good amount of beta glucans, which is a good functional ingredient for our gut bacteria. And then we also picked the yeast, which is Chloromyces maraxinus, which can grow on lactose, which is one of the sugar from dairy processing industry. So we selected our microorganism based upon certain parameters. And then we also identified a number of the feedstock for these studies. So we picked brewer spent grain coming from brewery industry, vapor meat, apple pomace, potato processing liquid, and grass. So we tested these microorganisms on these feedstock in order to see which strain can grow on these biomass. So of course, we performed different pre-treatment on each and individual biomass. We applied ultrasonication, we applied microwaving, we used enzymes and a combination of these approaches, and we basically recovered different sugar-rich hydrolysate, and we tested the sugar level in that. We also tested the protein content because that was in order to understand how much nitrogen sources is available or sources are available in these biomass. Because carbon and nitrogen, these are two primary, let's say, ingredients for microorganism to grow and to grow rapidly or to grow in an environment. So we wanted to understand what is the level of sugars and nitrogen present in these biomasses after a pre-treatment. So we picked different scenarios, different conditions out of these pre-treatment experiments, and then basically we grew. And we, of course, not all these hydrolyzed, they were not, as it is used, they were not as it is used as a media. So we topped it up because there was a less sugar as against the synthetic media. So we topped some of these hydrolyzed with additional nutrients that are essential for microorganism growth. So then we, once we optimized that media, then we inoculated that media with the right microorganism. and then we performed the fermentation in flask initially, and then we got the cell biomass. So if you see the difference in both, because here in the top flow chart we use Biosteatus, which is a fungi, and at the bottom we use Aspergillus, and we got cell biomass. So roughly the Biosteatus contains around 48% of total protein in their cell biomass, while in case of Aspergillus or IG, we got 38% of protein on a dry matter basis in the cell biomass. So technically, and all of that protein was produced, let's say in case of Aspergillus, it was produced within 60 hours, while in case of Biosteatus, it was produced within 6 days. So just see, you can produce that protein within a week, as against if you go back to traditional protein, so which is soybean. In animal nutrition, if I say it takes a year to produce that protein and a lot of processing, but here we produce a good quality of protein within a week, which is quite sustainable, and as against the traditional protein source. We also tested vapor permeate using yeast strain, and then of course the process was exactly similar. And then we got the cell biomass, microbial cell biomass, and we produced that cell biomass contained more than 40% of protein. And the another advantage of producing these microbial proteins, because not only the protein you are getting 40 to 50% protein at this stage, you are also getting other nutrients in the cell biomass, which are they contain good quality of lipids, and they contain good quality of fibers as well. So if you are using that cell biomass into any product, you are getting not only protein but other ingredients. There's a safety and issues as well, because these biomass contain a good amount of nucleic acid content. So we performed a step where we reduce that nucleic acid content that is recommended by the EPSAB. So we reduce because that is a bit toxic, I would say, for human consumption, but there are processes that reduce the nucleic acid content in the biomass, and then once you have less than 2% of nucleic acid in your biomass, that product can be used for human consumption. So this is the process prototype that we developed in this project. So there are different steps. We have what sort of steps were involved for making the final product, and what was the step involved in order to break down that matrix and removing the nucleic acid content and getting the final product. So this was our let's say a biorefinery process that we designed for utilizing BSE, which is coming from brewery spent grain or brewery sector, and we produce protein, and that protein can be used in animal feed straightforward, and it can also be used for human consumption. So the second case study, which is again very, very interesting case study, where we used potato processing side streams. So if we look at the potato processing side stream here in Ireland or anywhere, so this is the process that we use. So you grow potato, and you basically take off the aerial part of the potato, and you also take off the undersized or oversized potato, which has a different shapes. So you don't use them in commercial applications. And then when you perform washing, and then you do peeling, and then you send that for starch making, mainly here in Ireland. And then in that process, you produce huge amount of water, which contains a lot of nutrients that is coming from washing, as well as while making starch. And then, of course, you make different food product as well, like chips and other things. But if you see the whole process, you can see at 3 different places before you, when you are, let's say, if you're focusing on starch making, if you are a starch making company, you are generating huge amount of wastewater, which contains huge amount of starch in it. And so, in current scenario, if you are discharging that water, you have to do some sort of treatment that again costs a lot, and before you discharge that water into, so you have to reduce the BOD and COD of your water, so that give a extra cost to companies. And of course, you have a lot of peeling waste that coming from the potato, and a lot of green and aerial part of land that is unused at the moment. So in this, and if we see, as I said, you always have to look at whether you have that available biomass if you are working on the biorefinery. So we checked the potato production here in Ireland. So if you can see, so we per person roughly 80 to 90 kg potato consumption per year per person, that the amount of potato we consume here in Ireland. And if you see, we produce around 300,000 tons potato per year, which means we have a huge, potato production based considering the size of the country, as well as, of course, if you see, because a lot of that potato goes for starch production, and it produces huge amount of effluent, which if I put it in numbers, so you use 3 to 5 cubic meter water per ton of potato processing if you are working on a starch, which means you are producing a huge amount of wastewater that contains, of course, water, protein, sugars, and starch, which is a waste at the moment, and companies generally pay in order to treat that water before they discharge. So our idea was how we can utilize that wastewater, how we can develop a multi-product biorefinery, and that can also basically add certain revenues to those manufacturers. So this is the process that we developed, or we are working on it. So we have developed the full process for them together with our partners. So here, of course, we initially we removed the solid part from the water, which contained a big amount of the starch. So we recovered 6 to 7% of starch from that potato water through certification, drying, and we performed the nutritional profiling, and we identified the different nutritional protein content, starch content into starch, and that is one of the products that can be produced out of that let's say wastewater. And then the water, let's say if I come back to here, so you got the, after sanctification, you got the solid paste, and you also get the water. So this water basically it contains a lot of other like ingredient, which is like mainly the proteins and some sort of sugars as well, coming from starch, that is water soluble starch that is present in the water. So we performed some pre-treatment. Here we use ultrasound, different enzymes and chemicals in order to break down that starch into sugars, and it also releases the proteins that are bound with different other biomolecules. So again, perform sanctification, and then we produce a protein-rich fraction and a sugar-rich fractions from the water side of the fraction, and then that if I say here, the protein-rich fractions were further dried, and that is protein, that is a second product that we produce out of that water from the initial sample. And this protein is basically, it is a potato protein that is present in water that is coming from potatoes. Okay, so there's so that these are two things, starch as well as protein. They are, let's say, indigenous ingredient of potato. So it was present in potato, and we recovered these two products from the wastewater. Now, because there was another fraction left, which was a sugar-rich fraction, so we wanted to develop something for this. So we used, again, a microbial fermentation approach, where we performed, we were trying to produce a single-cell protein out of it. So we used yeast and fungi again, and then we used their sugar-rich fractions, and we topped it up with different nitrogen sources, and then we produce a microbial protein. We perform downstream processing, and then we get a single cell protein. But you have to understand one thing with this this single cell protein is completely different than this protein, because this was the part of potato, but this is a completely new protein based upon the microorganism. So this has a very, very different amino acid profile, and that protein can also be used in different food and feed applications. So if you see this biorefinery that we produce, which is a multi-product biorefinery, and it is creating zero waste, and the water that is coming in the end is completely has less BOD and COD, and you are getting three different product streams out of that water. So this is the refinery that we developed, or we are finalizing that biorefinery. Then the third one is the last slide. I would say I wouldn't take much time on Friday evening. So this is completely a different kind of biorefinery that we are developing, not for food, but for agriculture. So, like because we are a dairy based, I mean, we have a huge dairy sector here in Ireland. So, all of these dairy processing industries of plant, they produce huge amount of sludge and digestate and after the processing of milk and cheese. And apart from that, we also produce a lot of digestate through our wastewater treatment plant. And if you see, because the government's new initiative for installing number of ADs by 2030, of course, all these ADs is going to produce huge amount of digestate. And at the moment, we don't have any kind of specific policies for utilizing that digestate, because a lot of that digestate straightforward goes into our agriculture field. So, of course, it is all that digestate is not 100% safe, because you have after processing through AD, you're concentrating a lot of things, which means you might have a lot of depending upon the feedstock, you might have heavy metals or mineral load, or you might have different kind of microorganism or microbial load in your digestate. And if you're spreading it into agriculture field, which means you are exposing your natural microbiota of soil with different microorganisms, so which is which could be a challenge. So going forward, if we are going to produce huge amount of digestate, we should have another strategy. So this project is again funded by EPA and Department of Agriculture, Food and the Marine. So in this project, we are trying to develop a process or technology, so where we are using digestate as a feedstock in order to make some solution for that digestate, and on how we can make that digestate a little bit safer for application. So here we are we have developed a process, a pre-treatment process that not only basically minimize the microbial load in the digestate, but this process is producing high-grade agrochemicals from that digestate. And these agrochemicals can be straightforward. They are organic chemicals, that agrochemicals, they are biostimulant. So we are targeting biostimulant from these digestate because they are rich in carbon and nitrogen. So we are focusing on humic substances. They can be used as a biostimulant in crops. So we developed a process that is recovering the agrochemicals, especially biostimulant, from the digestate. And because this is a cascading biorefinery process, so initially, because our starting material is digestate, which is itself a waste, okay? And then utilizing our pre-treatment technology, we are producing the biostimulant, which is agrochemicals, organic chemical, and whatever is left after that, we are diverting that through a thermal treatment process using pyrolysis and hydrothermal carbonization. Through this process, we are producing, of course, some sort of energy, not in big quantity, but still we are producing energy. And we are also making biofertilizer in the form of biochar and hydrochar. So, and then we are also, because as I said, through pyrolysis and hydrothermal carbonization, we are producing hydrochar and biochar, and we are getting agrochemicals from this biostimulant from this previous process. So we are developing two products. One is, I would say, three products here. We are making a biostimulant. We are also developing a biofertilizers. And we are testing both product and their combination on different plant trial experiment. We are testing them on tomato plant. We are also testing on barleys in order to understand whether these agrochemicals can be used in these crops. We are hoping that this process can reduce at least 10 to 20% of synthetic fertilizers through these organic source of fertilizers. So that is what we are doing in this biorefinery concept. And if you look at it, and we are also performing the life cycle assessment of the whole process, and where we want to understand the environmental sustainability of the entire process, because there are a couple of energy intensive processes are involved in these biorefineries. So we want to understand whether our process is safe and our product that we are producing is again safe, as well as is competitive uh, as against the already available product in the market in this space. So I would say that's all. That is what we are doing at the moment, and apart from there are a few more processes that we are developing. So I would say, because in terms of the time constraint, I would say that's all. And if anybody has any questions, please feel free to drop an email. My email address is on the front slide. And if you have any questions, and I'm happy to answer. Thank you.
Thank you very much, Gaurav, for describing so much interesting slides. So slides were self-explanatory and very good. An excellent delivery. I really enjoyed, and you left me with no questions on the topic. Thank you very much. And you have covered a wide range of topic overall. You have covered the whole Irish bioeconomy resource and the technologies being used. So thank you very much once again. IBF team and the Rural Bio team is grateful for your time and contribution. Thank you very much.